Welcome, everybody. My name is Gustavo Mostoslavski. I am one of the co-directors of the Center for Regenerative Medicine. You will meet later Marianne James. She's the manager of our IPSL core. And we are really very happy to have you all here. This is the second year we're giving this course, and we were very excited to see that very quickly all the places got filled, clearly showing that there is a lot of interest from people to learn. We recognize also from our previous experience that there is a wide variety of backgrounds among the participants. So you will see that the class starts kind of basic in purpose, because in some of that, for some of you, may be too familiar, or you already know that. But we decided that it's important, and you will see also a lot of historical context for us, also how we got involved in the world of iPS cells. And this is supposed to be a dynamic class, not only because of the practice. You will get a lot of practice this week. It's kind of amazing. We are very proud. It was last year was a great class. The people really left happy, and now all of these people are using what they learned here. So we hope that this is also going to be the case for you guys. So feel free also to interrupt me and ask questions along the way when I'm presenting, and you will have plenty of time during the whole week to come and talk to me or to talk to the rest of the presenters. Tomorrow you will meet Daryl Cotton, who you probably know the name. Daryl really has been changing what we know about how to use pluripotent stem cells into the lung field. And a lot of what you're going to learn this week about endoderm differentiation and lung differentiation comes out of his lab and his research. And so you will meet him tomorrow as well. So today is our intro class, so is that okay? Anyone has any question about what's going to be the week? After my presentation, which is supposed to be maybe an hour, even less, Marianne will give you a few slides to try to tell you what you're going to be doing during the day and during the rest of the week. Okay? So this is a Center for Regenerative Medicine. And we created the center as a virtual center first around 2009. And you will see a little bit of how historically why we came up to be a center, why we decided to be a center. But there are, like right now, 6 independent labs working, and there are 2 more PIs joining this year, the CREM, and we are interviewing actually for a 3rd one. So we're clearly expanding and growing. And we moved into this space, this beautiful space, in 2013 as a real center. And as I said, there are 6 PIs working in many different aspects, but we kind of all center around induced pluripotent stem cells and doing different approaches into going into different lineages. And we are part of affiliated with different sections in the Department of Medicine and Pulmonary Center. I am part of the GI section. We have a faculty from the Hematology Oncology section. And because of that, we're also going into the SSS and different lineages. And we're going to talk about a little bit about that too. But today, I'm going to give you an introduction about, in general, pluripotent stem cells. And I'd like to tell you this. So this is, if you try to come up with a definition of stem cells, you can find many different ones. I like to define stem cells based on their properties. And I think this is probably very familiar to all of you. But we know that stem cells can divide without limit, okay? And it has a name that's called self-renewal. And also that they are not terminally differentiated. So that means that the stem cells per se cannot function as a mature cell with a defined function, right? When they divide, daughter cells can either become another stem cell or begin the process of differentiation. And this is basically really a key in defining a stem cell. And this is important because we like to be very precise when we define a type of stem cell. And this is important because it has actually implications. You know, there are in general, 4 types of stem cell defined by their potency. They can be totipotent, pluripotent, multipotent, or unipotent. And we'll talk about this a little bit more in the next few slides. But understanding the difference between these has a very important functional significance. Because, you know, for years, there are companies out there that they want to claim that you can use bone marrow cells to recover a defective pancreas. And that was based in very old studies that claim that you can do a bone marrow transplant and the cells has plasticity to be transformed into pancreatic cells or liver cells or brain cells. But we know that functionally that's not true. Bone marrow cells are a type of multipotent. That means they can give rise to different lineages, but of the same tissue, in this case, blood, right, the hematopoietic cyst. Anyway, so this is kind of the definition that comes out of these properties. So stem cells are cells that have the ability to self-renew indefinitely. So that's a key property of stem cells. They self-renew. So they give rise to more stem cells when they divide. But under certain conditions, they will start differentiating into all the different cell types of a tissue or an organ. Okay? So I like to see this always. This slide is a very, very old, basic slide, but basically shows what I just said, right? So the cell can divide and become either two stem cells, or under certain conditions start differentiating and making more mature cells that have now a specific function. Now, why are they important? Why we need stem cells? Why we care about stem cells, and why you are here today? Well, from the regenerative medicine point of view, they serve as the ideal source of cells during normal tissue and organ homeostasis. It's like basically keeping a very small pool or protected cells that in case we need them, then they will start dividing, differentiating, and repairing, for instance, when tissue is damaged. That also happens in case of a disease. And we hope that we can also use these stem cells to understand better the diseases and to find new treatments and therapies. This is what I told you about the potency and why this is important. And I'm going to make a 1 minute just stop here just to emphasize this. Because if I know that a stem cell is a multipotent stem cell and not a pluripotent, I cannot assign functions of a pluripotent stem cell to a multipotent stem cell. And the basic difference between these four types is if you look into totipotent, this is basically the egg. And it's a zygote. And that's the only one that can really have totipotent abilities. And that means that that egg can give rise to all the embryo and also extra-embryonic tissues. And that's the main difference with pluripotent stem cells. Now, pluripotent stem cell, you probably know already, the clear example of a pluripotent stem cell is the embryonic stem cell. And people have studied embryonic stem cells for many years now. The original description and discovery of embryonic stem cells in culture was done in 1981. And that was from murine cells. It has a beautiful story behind it, how people really found the conditions to grow these cells. And then I will talk about that also a little bit. And this is a perfect example of pluripotent. The one that we're going to see and talk a lot this week is, of course, induced pluripotent stem cells, which basically really changed the history of biomedicine. That discovered in 2006 by Shinya Yamanaka, who got the Nobel Prize. Multipotents are cells that actually still can differentiate into many different lineages, but within a certain tissue. The perfect example, the one that we have been studying for many, many years, is the hematopoietic stem cells. So again, this is important because we know the hematopoietic stem cells can give rise to the entire hematopoietic system, all the different lineages that are in the blood, but they cannot give rise to a liver or to a pancreas because they are multipotent and not pluripotent. Unipotent stem cells, these are a specific type of stem cell that in general can give rise only to one lineage within that specific tissue. And one example is the spermatogonial stem cells. They can only generate sperm. This is an important concept that you need to take with you. Now, embryonic stem cells. So I'm going to very briefly mention these cells. I think you're going to see a little bit more about this tomorrow with Daryl. But basically, this is the gold standard. So 1981, as I said, two independent groups found the conditions to take, if you take a fertilized egg and incubate it in vitro for a few days, they will form this structure called a blastocyst. And within this blastocyst, this group of cells here, it says ICM here, okay, the inner cell mass. They found the conditions to take these cells out of the blastocyst and put them in culture. And when they did that in the right conditions, they start forming colonies, and these colonies can propagate in vitro forever. You can freeze them down, you can thaw them many years later, and they will still now keep growing as colonies if you keep the conditions for pluripotent stem cell conditions. And those conditions were, at the beginning, they're not trivial. It took them years to find the right conditions. And we have evolved from those times. And you can see here that a bed of fibroblasts. These are mouse embryonic fibroblasts that are inactivated and serve as a feeder layer. That's were the only way that people could grow embryonic stem cells for many, many years. Now we do it differently. We can do it feeder-free. It has a lot to do thanks to companies that actually spend money and time to develop these protocols to make our lives easier, basically. So what we know, and I'm going to show real data confirming this, is that at any time, if I change the conditions, or if I take these embryonic stem cells, and I can do this in specific animals but not in humans, put them back into a blastocyst. If these cells are really pluripotent, they will give rise to all the different cells of the 3 germ layers, endodermal, mesodermal, and ectodermal cells. And we know that because, as I will show you in a few slides, we know that we can do that, and we know that these cells are capable of differentiating into all cell types. This is an amazing property. And people got into embryonic stem cells studying actually teratocarcinomas. They knew that there were these tumors that can give rise to all the different organs, and they thought maybe we can find a cell that is not tumoral, that is not transformed, that can still keep that property. And that's actually how they found embryonic stem cells. However, if we want to use embryonic stem cells specifically for regenerative medicine, there are some major limitations, and scientists have been known for this for years. Okay, so 1981, murine, and it took many years, 1998, for the laboratory of Jimmy Thompson in Wisconsin to discover the way to expand human embryonic stem cells in the right conditions. And they grow slightly different than murine, but it was 17 years of difference between the murine and the human until they found the right conditions. In any case, the first one is technical. So not many labs had access to eggs, especially human, and that was complicated. Not many people have the right skills because they couldn't learn this, because only very few labs could do really this type of work in the lab. So that's the first one. The second one, I'd like to mention ethical concerns, because it was actually a major limitation for a few years here, even in America. There were strong prohibitions from NIH to use to destroy new eggs to obtain new lines, new embryonic stem cell lines. So some laboratories found private funding to do that and keep creating lines. But that was definitely an issue that was up for discussion. The one that I like the most is this one, is immunogenicity. And that means the way that we can make embryonic stem cells from human embryos is mostly from donated embryos that are obtained from in vitro fertilization clinics. And that's perfectly legal. So couples that are in the process of doing in vitro fertilization, they can donate frozen eggs that are done in a case just as part of the therapy for research. So that's fine. They can actually donate the eggs for research. And that's how many labs obtain eggs to isolate the inner cell mass. But if you can make actually a tissue or an organ from embryonic stem cells and you want to use this for a transplant, you will have the same issues and the same problems within any transplant. So you will have the need for immunosuppression. Most of the time is for the lifetime of the patient, and with all the consequences of doing immunosuppression. So this is a real issue. And people have been thinking how to surpass that major obstacle for, at least for regenerative medicine. This is 4 examples of those, and I'm going to mention specifically 2, which are the most common ones. The first one is the one that we know, and you probably know, as somatic cell nuclear transfer, and commonly referred as cloning. And this is a technique, for instance, that was used to clone the first big mammalian, which is Dolly, that everyone knew. It was everywhere in the news. And the technique basically, it resides in taking a nucleus from a somatic cell. That can be a fibroblast from a skin. In that case, was from a mammary gland of a Dolly. So you isolate the nucleus, which is not easy. It's complicated. It's mechanically disruptive. But you can do it. And then you can introduce this nucleus into an enucleated egg. So you start with an egg too. But you remove the nucleus of that egg, and you put the nucleus of the cell that you would like now to have the genetic identity of. So that's why it's called cloning, okay? Or somatic cell nuclear transfer. And then you artificially activate that egg. And people use either chemicals or a current, an electrical current, and then these eggs start dividing. And in some cases, very infrequently, it's very low efficiency. In the case of Dolly, it was more than 266, I think, attempts to get one sheep. But then you can obtain a blastocyst with cells, and either you can isolate now these cells and make a pluripotent stem cell line, like I just mentioned, like human embryonic stem cell line, or you can try in animals to put that back into a pseudopregnant mouse and obtain a clone animal. Okay? So that's a technique of somatic cell nuclear transfer. For many years, people have succeeded with many different species. With humans, it was very difficult to do until only very recently that was finally successful in 2013. It was a little bit too late because by then we already been working for many years with induced pluripotent stem cells. So I'm going to jump directly to this one because these are less frequent, but these are basically slight modification of this original technique. And this is fusion. So people have shown, this was one major science paper from Kevin Egan's lab, that showed that if you fuse a fibroblast with a human embryonic stem cell, you can now activate some of the properties of the fibroblast in this pluripotent. So you can create a pluripotent line by fusing a fibroblast with a pluripotent stem cell. So something in the cytoplasm of this pluripotent stem cell has the ability to reprogram the nuclei of this fibroblast. Okay, and this is important of the concept of nuclear reprogramming. The problem is that the obtained cells in this fusion actually are R4N. They are tetraploid. So they have little real applications because this is a transformed line, has a multiple set of chromosomes, which is not really what we want. Okay? By then, people were thinking and dreaming about the idea of doing something different. And we're welcome to this world, Dr. Shinya Yamanaka. So Shinya Yamanaka came out of Gladstone in California, where he did his postdoc, and he learned a lot about how to do transgenic mice. He worked with transgenesis, and one of the genes he was interested in was called FBXO15. It's a gene that he thought it was very important for pluripotency. And he came back to Japan as an assistant, very young assistant professor. And because he knew a lot of transgenic mice, they put him as the head of the transgenic core in the institute that he came back. And he was deciding what to do, in part with his research, and also with he needed to create one transgenic. So the first transgenic he made was basically knocking out that FBXO15. And why this is important? Because he really thought that that gene was key for pluripotent stem cells to exist. And in addition, he was working into finding other genes that were important. And in 2003, he published a beautiful Cell paper describing basically NANOG. Now, many of you already probably have heard about NANOG. NANOG is one of the key core factors that are important, again, for the identity and the function of pluripotent stem cells. His lab described that in 2003 in a paper in Cell. And at that time, in that paper, in the introduction, he mentioned this issue of human ES cells raise substantial ethical issues, since human embryos have to be destroyed to generate ES cells. One solution to avoid such ethical issues is to generate pluripotent cells directly from somatic stem and other cells. So he was already dreaming about that idea. And he thought also that might be a cool project to bring students, because it was not easy for a young scientist in Japan to attract students. So he put an ad saying, we're going to try to make stem cells from skin, from somatic cells. And that's kind of a crazy idea, because a few groups have thought about it, and they all thought that it's going to be almost impossible. It's very, very, very difficult. You're going to need maybe thousands of factors to transform, to convert, to reprogram a somatic cells back in time to become a pluripotent stem cell. So this is the basic idea. So he actually did get a student, Takahashi, and they both come up with this plan where they look into all the databases of where were the genes that basically were always highly expressed in ES cells. Okay? And they come up with a list of 24. And this is the one that I mentioned before, FBX15. And we can talk more about that, why that was important in that time when they published the first paper. But that original list of 24, that's what basically they decided what's going to happen if we take now a somatic line, a somatic cell, like a fibroblast. And they specifically use first a mouse embryonic fibroblast. Then people can also show that it works with tail tip, a piece of a tail of a mouse like we did, and I'm going to show you that, and overexpress these 24 genes. And Takahashi did that experiment. In that original paper, he used retroviruses, individual retroviruses, 24 individual retroviruses, and infect fibroblasts of a mouse. And I'm already going to mention these 4, because he did it. He put the 24 genes, and suddenly, after a few days, he started seeing colonies. So he went to Shinya Yamanaka, and he said, Dr. Yamanaka, I'm seeing colonies. He said, no, that's impossible, because this experiment cannot work. So go and repeat. And he repeated, and he obtained again colonies. So he said, I think I'm seeing colonies. And they look like embryonic mouse stem cells. He said, wow, maybe I am believing that now you're getting something that looks like embryonic stem cell. But wouldn't it be cool if we know really which one of these 24 are important, are really key to make this reprogramming happening? So go back and repeat the experiment 23 times minus 1. So remove 1 at a time. And when he did that, he found 10 factors that seems to give still colonies. Okay, when you remove those 10, basically, you don't see the colonies. So he said, I think there are 10 that are important. So Shinya told him, okay, go on, repeat it again. 9 more times, remove 1 at a time of these 10. And he found 4 factors. OCT4, SOX2, KLF4, and CMYC, also known today as the Yamanaka factors. And it worked. It worked incredibly well. It's such a robust approach that the first paper came in Cell in 2006, and everyone immediately said either this is impossible or I need to test it. And basically worked in many, many labs. It's so robust that actually you take somatic cells, and it can be any cell, actually, not only fibroblasts. We are doing it routinely now from blood, and I'm going to show you a slide about it. And basically, they overexpress these 4 factors only, Yamanaka factors, and you end up with colonies that basically are identical to embryonic stem cells. And this was originally done with the murine cells. He got the Nobel Prize in 2012. This is 6 years after the original discovery. This is a very, very fast Nobel Prize, Nobel Award. This is me with Dr. Yamanaka. This is Daryl Cotton. You're going to meet him tomorrow. And this is George Murphy. He sits in that office next to me. And he's also a co-director of the CREM. We actually, the 3 of us, we founded and co-direct this center. And in 2009, we were in Barcelona in the ISSCR meeting. And that was 3 years before he got a Nobel Prize. But he was already a star. And everyone wanted to talk to Dr. Yamanaka. And he was just going around visiting the beautiful Sagrada Familia in Barcelona, and we saw him. And said, Dr. Yamanaka, can we take a picture of you? And of course, he said yes. I mean, he was a little bit stressed, they don't like much pictures there. But it was a great moment for us, and we were very happy because we were already working a lot with iPS cells. We knew immediately about the power of these cells. And he got the Nobel. You know who is this, of course, right? This is Sir John Gordon. So Dr. Gordon received the Nobel Prize because of his original studies published in 1962 about demonstrating that a nucleus actually keeps the capacity as a total, the entire genetic material is there. And he showed that in frogs. So he was able to show that he could take a nucleus from a somatic cell in a frog and put it and get basically clone frogs. And it took many years to show that that was true for mammalians, but actually that was the original major paper that came out of those studies, 1962. So it took him 50 years to get the Nobel Prize and share it with Shinya, who only did his discovery in 2006. So science is like that. And this is it. So I like this slide. I've been using this slide for many years now. I still like it a lot because it really summarized what was the big deal. Why all the rush, why he got the Nobel Prize. And why I mentioned before that he truly changed the history of biomedicine. For the first time, we can take now cells from anyone, from any individual. It can be a deceased individual with a particular disease, or it can be anyone. And just by taking a little bit of blood or skin fibroblasts, and people at the beginning, you were trying all kinds of things like hair keratinocytes, just taking one hair and see if we can get APS, which, by the way, can be done. It's not that simple, but it can be done. And by exposing the cells to 4 transcription factors, the cells basically change their identity to become pluripotent stem cells. Now, just to remind you, we don't have pluripotent stem cells in our bodies. They don't exist. It's very short window of time during development that we can access pluripotent cells. But any of us, that's it. We lost that window. So that discovery really changed that. Now, as a matter of fact, we've been working, for instance, with a disease called Lynch syndrome, which is a type of colorectal cancer, hereditary colorectal cancer. And we have a collaboration with Dr. Lynch, who is 88, I think, now. And he still goes to the lab every day. So he has the largest collection of Lynch syndrome patient samples, frozen, that he's been collecting since the 60s. And we asked him if we can collaborate. We would like to do iPSs from Lynch syndrome patients. So he sent us some vials that he froze in one of them was 1982. Of course, the patient died already. But we have this vial of frozen cells that is almost 30 years now. We made iPS cells from that, from those frozen cells that have been in the freezer for 30 years. So it's an amazing, amazing, powerful technology. So we can do this. And this is just now a routine, and you will learn this week. Okay, we can really now routinely make iPS cells all the time from a very little sample of blood from any individual. Now, we've been using this for this. So we've been learning, and this is thanks in part to what we've been doing scientists for many, many years with embryonic stem cells. So similar protocols now have been used for iPS cells as well to differentiate these cells into specific lineages. Now, we know that these cells can do it. We know that, and I will show you how. Now we need to learn how we do it in vitro, the same way that these cells can do it in vivo. Okay, and this is what we've been spending a lot of time to optimizing these protocols. You will learn a lot about endoderm and lung. That's why many of you came to this course. As I mentioned, Daryl's lab has been really putting a lot of effort and publishing major papers in how to do that, actually, how to obtain specific lung lineages out of pluripotent stem cells. And the idea, if we can do that, then of course we can study normal embryonic development, but this is not new because you can do that with embryonic stem cells as well. But now we can do really disease modeling in a way that is unprecedented. We couldn't do that before. People use transformed lines or animal models, but really accessing primary human cells. If they come from a patient, they already have all the genetic content of that patient. And I can now try to see if I can study the disease by obtaining the tissue or the organ that is affected in the disease in a Petri dish. And of course you can do drug screening, because now you can test many, many drugs. It's much cheaper and faster and easier than that's doing in a clinical trial. And one day, we hope also that we will be able to create so good tissues and organs that we can do also cell therapy for replacement. And there are many diseases and examples. Here we're studying actually 15 diseases now. And we try to do diseases that are related to the population of this hospital that we are affiliated with. Now, I need to mention the STEMCA. I cannot not do it because this was one of the major discoveries that came out of my lab. So when I joined BU in 2008, that was a year after the human iPSs were published, actually a few months after that. And I was thinking about having a new way to do it. As I mentioned to you before, the original protocol used individual 24 individual retroviruses, and they integrate. But because they integrate, which is a good thing if you want long-term expression, they can be risky because you have now many multiple integrations all over their genome. And that can cause problems, especially because these genes are not genes to be played with, including c-Myc, which is a known oncogene. And indeed, what happened is when they used these iPS cells that were generated with the original retroviruses and made mice out of these cells, 40% of these mice developed tumors. And when they check the tumors, they will all have reactivation of c-Myc. So people knew that we should find better ways to do it. The way my laboratory did it, we put all 4 genes together in a polycistronic construct, and this was a lentivirus, and we call it STEMCA, the stem cell cassette, and worked really, really well. By doing also introducing, and this is the original experiment that we did in the lab. So by then I was already collaborating with a good friend of mine, Konrad Hochedlinger, who is at MGH. He has his lab in MGH. His postdoc was Matthias Stadtfeld, also a very good friend of mine. And they just created this mice, a knock-in mouse that has SOX2 GFP knock-in. So these mice, basically, their cells express GFP under the control of SOX2. Now, SOX2 is one of the 4 factors, but also is a gene that is very highly expressed in pluripotent stem cells. So one easy way to know if your cells that you're obtaining, and today, of course, we don't even need that because we just know how to see the cells. But at those days, we're not that straightforward. So if you have a marker that can tell you you're in the right direction, you're getting cells that seems to be reprogrammed. So that was why we got these cells from them. And they also have an M2 RTTA, which is a factor that you need if you want to have induction with tetracycline or doxycycline inducibility. So we asked them, can we get some cells from these mice? And they say, sure. So they cut a small piece of the tail, and they send us that small piece of the tail. And we grew fibroblasts out of that tail tip, okay? And after we expanded the fibroblast, we exposed them to the STEMCA. In this case, it's the version that was inducible, but we made all kinds of versions, inducible, constitutive, mouse, human version, with 3 factors, with 4 factors. And we can talk about that if you're interested about the differences between using 3 or 4 factors. But we basically infected the fibroblast, put them in media, ESL media, and wait. And indeed, we got colonies. So after we have colonies, you can pick the colonies, like expand them, and let's see if they grow as pluripotent stem cells, like embryonic stem cells, and they really look like identical to embryonic stem cells. So that was a big day for us. And what you can see here in the background is the feeder layers, and these are the colonies. And if they are pluripotent, they should be green, because you remember this is a knock-in of SOX2 GFP, and they are green. Now, how can we know that they are pluripotent? Well, there are different ways to test for pluripotency. And I think also there I may mention a little bit more of that tomorrow. But one of the ways, basically, let's see what markers they express. So this is a classic panel of markers we can test. One of them is alkaline phosphate. This is for murine. It's different from human. SSCA1, another marker. And here there is a list of markers that are, this is just common PCR to see that the cells, the different cells express these markers in contrast to the fibroblast. And we did both. We did a constitutive version. We did an inducible version. And this is just something, it's interesting. At that time, it was very important for us to show that our STEMCA only integrated a few times in the genome. As a matter of fact, in most cases, most of our colonies has only 1 integration. And this was important because in contrast to the previous protocol where they use multiple retros and they have an average of 30 to 50 integrations, we can do it with only 1 integration. And if you have one integration, it's very easy to remove it. So we basically built, and this is just showing that the average was 1.5 for the constitutive, a little bit higher integration for the inducible. This is just some of the properties of our STEMCA. At that time, we obtained an efficiency of around 1%, which was very, very high for those days. Okay, it's around 50-fold higher than was previously reported. Now, you can say, well, it's really, does that make any difference? It really matters if it's 1% or 0.1%? Well, I can tell you. We have been preaching what we call open source biology. And you can read about it in our website, in the CREM website. And that means complete unrestricted sharing of reagents and ideas that are coming out of this center. We truly believe in that. We really believe that that's a way to move science forward. So at that time, people start, when we published the original paper of the STEMCA, people start asking for us, can we share the STEMCA? And we did share it. Actually, more than 650 labs in the world received the STEMCA. And that's where the difference happened. Because the retroviruses were not that efficient, and some people had trouble getting colonies when using the retroviruses. But they used the STEMCA, and the efficiency was really higher. And they basically finally saw colonies appear, and they were very, very happy. And also because we share it so much. In addition, with the inducible version, you can also test what people are already having showing. So how many days do you really need the expression of these exogenous factors for reprogramming to happen? And the experiment is very simple. We basically expose the cells to either 1, 2, 3, 4, 8, 12, 16 days, and then remove doxycycline and say, do you see colonies? And are they green? And when you do that, what you see basically is you need at least 8 days. At least 8 days of induction of these exogenous factors to make the cells now start producing their own endogenous factors to become stable pluripotent. Okay? It's even better if you even keep more time doxycycline. Now, these days, this is really not relevant. We know how to do this really well. It becomes, as I said, almost a routine process. But on those days, this was very important. Now, it's interesting from a biological point of view, because I can ask you, do you think that 8 days is slow or fast? Well, you know, you can say, maybe it's fast, 8 days. You're really changing the identity of a cell. Well, for biologists that were working in those days with pluripotent stem cells, they thought it was not that fast because somatic cell nuclear transfer, if you do this transfer of a nucleus into an egg, change of identity happens almost in a day. Now, how do we know? Because people did experiment where they have a knock-in of OCT4 GFP, so they can see how quick GFP start being expressed. Now, that's really not exactly the same because it's not full reprogramming, but at least it's the activation of a gene that we know it's supposed to be expressed only on pluripotent stem cells like OCT4. And they really saw GFP starting to appear in this somatic cell nuclear transfer experiment almost 24 hours after the transfer. So it is very fast. This takes at least 8 days. So it's a little bit slower. And we know now people and many labs have spent years understanding exactly why this takes a few days. And mainly basically is trying to surpass epigenetic barriers. The epigenetic obstacles of transforming or changing the identity of a cell from a somatic cell to a pluripotent stem cell. Now, markers are not really a proof of pluripotency. I want to see functional proof, which is the definition, right? Can we show that these cells can differentiate into different lineages? One crude assay is called teratoma assay, where you take your cells, and in the case of mouse or human, you can inject them into the back of an immunocompromised mouse. And when you do that, what happens with pluripotent stem cells that have blood supply, so they have food, they can grow, they start spontaneously differentiating into all the lineages that it can actually make. And that's why you may make a tumor. It's a benign tumor called teratoma. Okay, you know where the word teratoma comes from? Teratos, monster. So that's how it was defined, because these are very ugly tumors. They can have hairs and teeth and bone and cartilage, and all actually lineages from the 3 different germ layers. Right? And we did it with the constitutive and inducible and show that we can obtain teratomas. But again, I want to see more. And one great assay, and we can do this for mouse cells, but not with human cells, is what happens if we inject the cells now back into a blastocyst. And you can take this blastocyst and inject it back into a pseudo-pregnant mouse. If our cells are truly pluripotent, they should contribute to all the tissues and organs in that animal. Okay? So we did it with the inducible stem cap, and this is one example. So this is an embryo, and this is an experiment we did in collaboration with Matthias Statzfeld in Konrad Hockettinger's lab. They did injection, we provided the cells, and he gave me a call. It was a Saturday afternoon, and he said, Gustavo, I think you need to come to the lab right now because I'm opening the embryos, and you may like what I see. Now, as you remember, we started with a piece of a tail of a mouse that was SOX2 GFP. Now, SOX2 during development is also expressed in the neural ectoderm. So now we have a marker to see if our cells are contributing to neural ectoderm. And this is what he saw. Beautiful contribution of GFP. Every single green cell there needs to come from an iPS. Now, if you see this in the neural ectoderm, that means that almost everywhere there should be contribution. In this case, most of these embryos is coming actually from our iPS cells. Now, these are chimeric mice. When you mix in the blastocyst your cells, it's already there. That blastocyst contains already endogenous ICM. So there is a chimerism happening, and you will see how that looks. But you see that picture there on the wall, out there? So this is not just one embryo. It was a very robust effect. At that day, we isolated, I think, 12 or 13. Most of the embryos have contribution from iPSs. So I called Daryl. I said, Daryl, you need to come to the lab right now, because our cells are pluripotent. This is it. This was the final proof that what we were obtaining with the stem cap from a piece of a tail in a mouse, we can now make an entire animal out of this piece of a tail. That's the power of iPS cells. It's really incredible. Now, we also did some sections and confirmed that the cells, the green, were really there. And this is how a chimeric mouse look. So you do a trick. You put your cells that come from a background that in general you can choose, but in this particular case was a mixed background. It's called black agouti. So it could be either black or agouti, brown, dark brown. And you put it in a blastocyst of an albino mom. And then you see, if you obtain this is true chimerism. Okay, that's biology, that's the best. Now you can go farther, because there were some proof that some cells that are pluripotent in that regard, they cannot contribute to the germline. And there were some defects in the cells that apparently they cannot contribute to the germline. What that means? That means that if I see here chimerism in the skin, and I claim that the same chimerism is also happening in the internal organs, the cells should also contribute to the germline, to the sperm or to the ovaries. And if that's the case, I can take now one of these mice, in this case was one male, and put it again with a white girlfriend. And again, this is a trick, right? Now my cells, again, are either C57 black agouti, and I'm putting now, again, with a white albino mouse. Now the skin color is allelic. So this is a chimeric mouse. That's why it's mixed. But here, if mice are born and my iPS cells are contributing to the germline, I should now see either black, agouti, or white. And this is what happened. Biology. So there you go. Now we obtain an entire mouse coming from a germline transmitted. This is full pluripotency. Okay? Guys, this is a very important concept. So it's important that you understand. Any questions? Okay. Now, at that time, it was very important. People complained that we were using a lentivirus that also integrates. So it was one integration, but it was an integration after all. And just to let you know, the risk of having an integration, it's called insertional mutagenesis. So you, by making that insertion, you're risking that your insertion may come next or close to a gene and may activate genes that you don't want to be activated. In addition, you are basically putting now 4 exogenous transcription factors, some of them are known oncogenes, there as an extra copy in the genome. This is not something good to have. So at that time, we designed a strategy where we introduce the LOXP site. And the way it happens is because it's here, and we're not going to discuss this further, there's no need to, but when this lentivirus basically does reverse transcription, it creates a copy of the 3' to the 5'. So now we end up with this vector that is entirely flanked by LOXP. And LOXP sites are short sequences that can be recognized by an enzyme called CRE. And if you expose now the cells that have an integrated version of the STEMCA to CRE, CRE can recognize these sequences and make an interesting biological process of looping and excising. And you end up at the end basically with an almost seamless iPS cell. And I say almost because you do retain around 212 base pairs from the original STEMCA. We like to believe that this is good because you can tag your cells. Basically, you know when you claim that you're having a cell that comes from your cells, from your iPS cells, you can actually even PCR that fragment. So it's a tag. In the gene therapy field, that was always very important. So we kind of like that we have that small piece still there. But you don't have any more of the factors, the exogenous factors. So when we make iPS cells with our STEMCA, in general, we excise the STEMCA. So it's not anymore there. There is also an important biological reason for it. We found, especially in the mouse cells, that if you keep the STEMCA, the cells have so high levels of these pluripotent factors, they have a hard time coming out of pluripotency. So it's not easy to make them differentiate. In the human cells, they do silence more. So the lentivirus gets silenced. So after reprogramming happens, there is no much expression coming out of this lentivirus, but we still decide it's better to remove it. So in general, we always try to excise the STEMCA when we do the STEMCA reprogramming. You will see, I think this week, STEMCA reprogramming or Sendai reprogramming?
Parts of Sendai.
Sendai. So we've been actually transitioning to using, there are many other methodologies. I will argue that today, most of the world use one of 3, either the STEMCA, Sendai, many groups have been using Sendai virus. It's a non-integrating virus that can have very high levels of expression, so it works really well for reprogramming. And some groups like to use episomal, the original episomal technique described by Jimmy Thompson. I think I didn't mention that in 2006, it was the original Cell paper by Yamanaka. His lab, a few months later, together with 2 labs in America, Rudy Jenisch and his student, that was an independent investigator, Konrad Hokedringer, that I mentioned, they do both publish together with Shinya an improved version of that original Cell paper, confirming that these cells were really truly pluripotent. And in 2007, 2 papers, Yamanaka and Jimmy Thompson, showed that human iPSs were also possible. And they use slightly different techniques. The Thompson lab used 6 factors instead of 4, and they use episomal delivery. And there's some labs that are still using episomal. Anyway, we also move into the human arena. That was all mouse. We basically changed the factors from murine to human factors. The truth is, they actually are interchangeable. You can use a mouse factor and make reprogramming of human cells because the factors are so conserved that the function still works. But we still made the human version of the STEMCA, and it worked really well. Again, they express all the markers. They are alkaline phosphatase positive. They were, of course, important, very important, chromosomally normal and stable. And they also have like single integration, so you can excise it. And because of our open share approach that I mentioned to you, we gave the STEMCA to everyone. And then many papers start coming out actually using the STEMCA. I like to put these two for a couple of reasons. So this is a cover in cell stem cell. At that time, 3 independent labs showed that you can do reprogramming from peripheral blood. So that's a nice picture. And we were together with Jenisch, we collaborated with one of these 3 groups. That original protocol actually was not very efficient. They used lymphocytes and it was extremely low efficiency, but it worked from peripheral blood, even from frozen blood. From those days, we actually made an improved version of the protocol, and now we can really do it from very little amount of blood, which you're going to see today. And this paper actually came out of a Catherine Plath lab in California, and she did a comparison at the genomic level. So she compared transcriptome expression of iPSs that were made either with individual retroviruses compared to the STEMCA, compared to ES cells. And she showed actually when you're using the STEMCA, they were transcriptionally much closer to true ES cells compared to the one made with individual retroviruses. So the conclusion basically says that the polycystonic delivery and extended culture brings human iPS cells transcriptionally closer to human ES cells. So it not only makes it easier, faster, but also makes cells that are better in terms of their transcription identity. Now, in those days, it was very important to convince everyone that when we say that iPS cells are similar to ES cells, they're really similar. Now, it's funny how scientists think and work, because in the first, if you look at the titles of the papers in the first 2 years after the original discovery, iPS cells are highly similar. Induced pluripotent stem cells are indistinguishable from ES cells, comparable, morphologically indistinguishable. I mean, we were pretty convinced that these cells were where we wanted to be. But then I guess scientists get bored. I don't know. They start looking for more challenges. So then things start changing. So the next 2 years, induced pluripotent stem cells are distinguished. So indistinguishable and now distinguished by gene expression signatures. They are prone to genomic aberrations, aberrant epigenomic reprogramming, somatic coding mutations. And then this paper came out saying that actually there is mosaicism in early passage. And if you expand the cells enough, they rapidly actually select against mutations. And you end up actually with a stable line that has no mutation and grows almost identical to ES. So this is one example. We now know better. There are really almost no differences. When you do full reprogramming and you wait a few passages so the cells become stable, the cells are basically identical. And now we have many, many studies to confirm that. In all levels, they do epigenetic studies, transcription studies. They really are identical to ES cells. Okay? Now, I think I put one slide of this. This is again from Conrad and Matthias Stadtfeld's paper. This is a Nature paper that at that time made a lot of noise. They tried to compare lines, and they found that some of the murine cells, murine iPS cells, could not contribute to what is called tetraploid complementation. Tetraploid complementation is a variation of obtaining germline transmission, but you use a blastocyst that actually cannot contribute to the animal. It's a defective one. It is the time that you actually can capture the blastocyst. And by doing that, if you obtain a mouse, it will all entirely 100% come from your iPS cells. So it's the most stringent test for pluripotency. Because there are certain lines that even though they will contribute to germline, they will not contribute to tetraploid. So this is really the most stringent test. And when they did that, they found that there were certain iPS cells in murine cells that could not contribute to tetraploid. And when they tried to compare those, what they found is a very small actually group of genes, all in the chromosome 12, that has aberrant imprinting. And that aberrant imprinting basically changed the expression of these few genes, and that's apparently what made these cells to be not as perfect as the ones that had levels of these genes the same as ES cells. Now, I mention this because at that time this was a very important paper. It was not really found in many lines, and it was also not found to be the case in human cells, so people kind of don't use that anymore as a reference. What about directed differentiation, which is one of the reasons you are here today? Well, if you look into PubMed, you will basically find out almost any lineage has been created from these cells. Some of these, and there are different types of neurons, endoderm to liver, vascular, pancreas, bone, ear hair cells. This is a great paper, actually. Retina, there are several papers, retinal epithelium, intestinal organoid, anterior foreguard, lung and thyroid, many of them coming from our own creme, cardiovascular, and many other things. George does a lot of blood here in the creme, and he likes red cells and megakaryocytes. I've been doing some as myself, some mielodendritic cells, and we're trying to get T cells. And so you can really find almost any cell now coming out. So clearly we know that these cells have the capacity to do it in vitro. Some of these protocols are robust and work well, some of them not that much, and we are trying to make them better. This is just, I don't expect you to read this very busy slide, but really it was a fever, a craziness in the first 4 years about who can develop a better approach, a better methodology. And when we finally thought that we have one approach, and now we come out showing that there was a better approach, a better technique, a faster one, or a safer one. The truth is that there are many out there, and most of them now are not used. As I said, I think that most of the world uses one of the three that I mentioned. Here, so you can find probably somewhere there, Mostoslavsky. That's when we publish our STEMCA protocol. And this is just a table. If you want to compare all the different methodologies, and some of you have interest in knowing, I can tell you a whole class about it. You know, there was a time when people publish episomal vectors. I say this is good. I don't know if I put here the transposon. That's the piggyback. So there were 2 Nature papers back to back about piggyback. And that was the best ever. Everyone said, oh, that is it. Now people are going to start using piggyback. And it didn't happen. So you can ask, why not? It was good. It was what they call seamless reprogramming. Meaning you can put a piece, in this case with the reprogramming factors, and then by exposing to a transposase, it will remove it completely without leaving a single base pair of difference. So it is really great. But it didn't happen. Why most of the world is not using piggyback? Well, I will argue because they were not that interested in sharing that reagent. And that's a real practical reason. And you can see, I mean, we are here 6, 7 years later, and most of the world is not using piggyback, even though it worked. And there are a few other examples. I mean, people have published for using recombinant proteins. No one can do that. So 2 labs publish, big papers. No one can do that. No one can reproduce that. But it's published. And of course, it's ideal because, of course, you don't have any problems when you put just proteins, right? So it's ideal if you can do it. Anyway, and it's fair to say that Senda is really well. It's one great technique, and most of the labs are using Sendai bars. Anyway, this is it for the theoretical part. Okay, I hope you learned some of the historical aspects of why we got into iPSS, why we are so passionate about iPSS. I think Mariana gave all of you a folder. You will have there a small booklet that contains almost all the protocols that you're going to actually learn this week.